Yes, good morning to all. In a last lecture, we started with this meiosis, the first round of meiosis, the reduction division. In that, so far we have discussed up to prophase 1, the one in which the five substages, leptotene, zygotene, pachytene, diplotene, and dikinases. That much we have completed. Let me show you what we have done so far in this meiosis. I think this is done, isn't it? We saw that how homologous chromosomes, they started condensation, they carried out synapses formation, further getting more close to each other and forming synaptonymal complexes. In each such complex, they crossing over to place which ended up with recombination. Then this chromosome started moving apart. Within each synaptonymal complex, the two homologous members started moving apart, getting physically separated except the points where they had brought about the exchanges. We call this as chiasmata. And in a diakinasis, even they separate at chiasmata. So terminalization of this chiasmata in a diakinasis. Important aspect of the diplotene that many of the vertebrates in their females, as and when oogenesis occurs, which is nothing but the meiosis for the formation of ova. Many of them go up to this stage, diplotene, and arrest this meiosis process entirely. And that can remain arrested for months or years to come. And later they resume it as and when required. So that's a peculiarity of diplotene. Dikinesis, as you know, apart from this chromosomal events in a cytoplasm, also that spindle, meiotic spindle formation, as well as nucleolus and nucleolus disappearance, nuclear membrane disintegration. These are the events which take place, which will lead to transition of these prophase 1 into the metaphase 1. Let's talk about our next topic, metaphase 1. In metaphase 1, you can see the meiotic spindle is already established. The developing microtubules will go and attach to this chromosomes as usual. But the pattern is little different. As you can see, pairs of homologous chromosomes which have already completed their recombination, such pairs are getting aligned on the equatorial plate. On this equatorial plate, it's not the single chromosomes, it's the pairs of homologous chromosomes which are getting aligned over there. If you remember, if it was a normal metaphase, it would have the cell in which individual chromosomes would have been arranged like this, isn't it? But here it is the pair of homologous chromosome. This would have been the normal metaphase in a mitosis. This is the metaphase 1. So in meiosis, this is a speciality that in the metaphase 1, the bivalent chromosomes, bivalent, paired chromosomes, they get arranged on equatorial plane. They get aligned on equatorial plane. Had it been a human cell passing through the metaphase 1, if this is 1, this is 2, how many such pairs would have been seen on equatorial plane? 23 pairs. Exactly, Gaurav, correct. And you can see that for each pair, look at this each pair. Microtubule comes and attaches to the centromere of chromosome only from one side, one pole. These are the microtubules coming from the opposite pole. They attach to the pair of homologous chromosome in such a way that one microtubule attached to one member and opposite microtubule attached to another member. That's the way the attachment takes place. Now, as usual, what happens in NF is the shortening of microtubules. As you know, these microtubules, they undergo the process of what? Shortening. Now, when they undergo the process of shortening, what is likely to happen? These two members, the homologous chromosomes, they will be migrating towards the opposite poles. You see, homologous chromosomes will separate, each with its two sister chromatids. Can you make out one centromere and its two sister chromatids? It is been pulled towards its respective pole. Have a look at it. The homologous chromosome, each with the two sister chromatids and centromere, they separate. One moves towards one pole, another one moves towards another pole. That's the reality. Now you see, had it been the normal, mitosis, the two sister chromatids would have separated with their centromere. Am I right or wrong? Just check out this, which can make you understand further. If this was supposed to be, first of all, metaphase, so like this, individual chromosomes would have been present at the equatorial plane. Okay, but there it is, a pairs of chromosomes. And another one, if I mention, no, wrong one has come. 1, 2, 3, and 4. This is the one. Have a look. For each chromosome, its two chromatids are getting separated, each with the centromere, and each one is moving towards the respective pole. Instead of that, here, if you compare, what is moving is an entire chromosome, one member from the given pair, along with its centromere, is heading towards the respective pole. Further, the telophase, as usual, these chromosomes which are collected at each pole. Can you tell me how many chromosomes will collect at each pole after the end of the anaphase 1? If it is the human cell passing through this anaphase 1, completed anaphase 1, Gallav, correct, 23 chromosomes. Shreya, wrong. 2 is wrong. How come 2? 23 pairs were here, for example. 1, 2, 3,
सर वो
4.
3 लिखना रह गया था गलती से.
Acha, okay, sir. So it should be 23 entire chromosomes here and 23 entire chromosomes at the other end. Aisa. And as usual in telophase, you know the things very well. Nuclear membrane formed around these chromosomes collected at each pole. nucleolus shall appear, chromosomes will begin the decondensation, the spindle is dissolved, cytokinesis will occur, and a pair of daughter cells is formed, which is called as dyad of cells. Excuse me. It is called dyad of cells. The two daughter cells come into picture, come into existence. That is the telophase 1. See, in overall context, we have completed up to here, if you have realized. This was the meiosis 1. Prophase 1, metaphase 1, anaphase 1, telophase 1, and it is over. So that is what we have learned so far, meiosis 1. Now, in this meiosis 1, which we have completed, this is what we have completed, meiosis 1. Cell entered from this interface in prophase 1, metaphase 1, anaphase 1, telophase 1, and has divided into two daughter cells. This is the cytokinesis 1, and this is called as dyad of cells. With this, meiosis 1 is accomplished. Any doubts? In prophase 1, homologous chromosomes synapsed, crossed over, created recombination, separated except the chiasmata point, and later even terminalized the chiasmata. These are the events associated with the prophase 1. In metaphase 1, the pairs of homologous chromosomes aligned at equator. In anaphase 1, the two members from each pair got separated. The two homologous chromosomes, excuse me, in each pair, the two homologous chromosomes, the two members from each pair, they got separated and headed towards respective poles. Further, in telophase, the two daughter nuclei came into existence. The spindle was, excuse me, spindle was disintegrated, and further in cytokinesis 1, the two cells got separated. They would be called as dyad of the cell. Now, immediately they will enter into what? After completing this cytokinesis, meiosis 1, immediately they would enter into what? Galau wrong. Yeah, interphase. What is the name of this short-lived interface between two meiotic divisions? What's the name given to this short-lived interface between the meiosis 1 and meiosis 2? Hey, correct. Interkinesis. That is termed as interkinesis. Good. And can you tell me how is it different from the interface, a typical interface? Is there a student who can explain the main difference between a typical interphase and the interkinases? A typical interphase between two back-to-back mitotic divisions in any cells, whereas interkinases, which is between two back-to-back meiotic divisions, meiosis 1 and meiosis 2. How is it different from the others? Not a single one, not a single student who is capable of replying to this question. Gallo is the one. Yes. Gallo, correct. The DNA replication, which is a part of a typical interface, G1S, G2S is the one.
Can I try?
Hello?
Sir, can I try?
Can I? Hello?
Hello, sir, can I try answering?
हाँ बोलिए.
Sir, interphase is a period when the chromosomes double, that is DNA replication takes place, while interkinesis is a period between telophase 1 and prophase 2, where DNA replication do not take place.
Okay, correct. So basic difference is typical interface shows DNA replication. The interkinesis does not show DNA replication. Okay. Another one, typical interface is comparatively long, whereas this interkinesis is typically a very short one. It is a short-lived stage. So short-lived stage, and then cell will again go back to its second round of division within the overall meiosis. This is called meiosis 2. This is termed as meiosis 2. And in that also there are typically 4 phases: prophase 2, metaphase 2, anaphase 2, and telophase 2, and cytokinesis. If each one will give rise to 2 cells, so the other one will also give rise. So altogether, how many? 4 cells will come into existence, and that's gonna be called as tetrad of the cell. If the previous one was known as dyad of the cell, this is gonna be called as a tetrad of the cell. Another aspect that this is more like a mitosis. This is more like a mitosis. Whatever chromosome number is here, same will be there in the daughter cell. So we call it as equational division. We call it as which one? Equational division. आप देखो meiosis के अंदर ही 2 टाइप के division included है. Check out this thing. This was a meiosis 1. It was a reduction division. Because chromosome number is reduced to half. देखो cell में 2 chromosome है, यहाँ पे 1 है. So reduction division. This is called as meiosis 2. अब देखो यहाँ पे एक क्रोमोसोम है, डॉटर सेल में भी एक एक क्रोमोसोम है. ये सिंबॉलिकली है. ऐसे के 23 पेयर्स होते, इधर 23 23 इधर 23 23 23 23. This is equational division. Remember. So within one meiosis there are back to back 2 divisions. This much is in a real sense the reduction division. This much is actually a kind of equational division. Have you all got the view, clarity on this aspect, the meiosis 2, what it is all about? I repeat the things which I said after the meiosis 1 and before the meiosis 2 beginning. A short-lived special interface which is without DNA replication. It is called interkinases that takes place between these two. This one, if meiosis 1 was reductional division, this one is called equational division. In a previous one, chromosome number reduced to half in daughter cell. In this one, chromosome number shall be maintained same as in mother cell. Same number of chromosome in daughter cell as in mother cell. At the end of the meiosis, if a dyad of the cell was born, at the end of the meiosis, tetrad of the cell, daughter cells, is born. Remember, typically in this one also, there are the four phases, that is prophase 2, metaphase 2, anaphase 2, and telophase 2, as usual. Let's look at these subphases. See, here you can make out with this meiosis 1 got over. Now would be meiosis 2, in that only one cell meiosis is shown. Same will happen with the another cell also. And there you can see the four cells coming into existence. So now what we are going to look at is just one of the daughter cells fate. Same would be there for the other daughter cell also, which is born at the end of M1. How many have understood this? Are you all clear with this or not, what we are learning now? चलो इंडिविजुअल एक डॉटर सेल का meiosis 2 देख लेते हैं. See, it's almost like a typical mitosis like pattern. The only thing is these chromosomes, if you see carefully, they are not ordinary chromosomes. They are the chromosomes which have already brought about what? Which phenomenon? What's the speciality of these chromosomes? Exactly, recombinant chromosomes. They have brought about recombination. Remember. So then, as usual, the things happen with them. Prophase 1, chromosomal condensation, nucleolus disappearance, nuclear membrane disintegration, spindle formation progresses. Then in metaphase 2, if it was a prophase 2, what we learned, this is the metaphase 2, where these recombinant chromosomes arrange themselves on an equatorial plane individually, each one centromere at the equatorial plane, and two chromatids are hanging. Each one is attached with microtubules coming from opposite poles, both side poles, one from here, one from here. So that's the way the arrangement happens. Then in anaphase 2, the shortening of the spindle fibers, microtubules, and when they get shortened, they split the centromere here and separate the two sister chromatids, one of them with the recombinant stage, another one without recombination. They get separated. The chromatids will be heading towards the poles over there, respective poles towards which they shall be migrating. And finally, in telophase, chromosomal decondensation, nucleolus appearance, nuclear membrane formed around chromosomes collected at each pole. Spindle should be disintegrated, and gradually through cytokinesis, two daughter cells come into existence. If this is the fate of one of the daughter cell generation 1, these are the daughter cell generation 2. So same happens with the other daughter cell also, D2, D1 rather. And there also two more cells will be formed. So altogether 4 cells will be born at the end of this entire process. The tetrad of the cell comes into existence at the end of this entire process. Here I conclude the meiosis. If you have any question, I give you a time, 1 minute. Think over it. Anything that you feel worth asking, please ask. Okay, it seems no one is raising question and repetition request, so it seems you have understood. If not, again, I will be sending the recorded video. Please go through it. It's a very simple once you have understood the meiosis one. Rest of the content is quite easy. We move to the last point related with the meiosis, and that is significance of meiosis. That is our next topic, significance of meiosis. See, first point, if organism is diploid and wants to maintain its diploidness, so when it is reproducing, the gametes that it is having, which are actually supposed to fuse with each other and form the zygote, they themselves should have been haploid. The gametes are supposed to be haploid. Because it's the same diploid zygote that will make a diploid body of the organism. So species chromosome number will be maintained. Our grandparents, their chromosome number is 2n. Our parents' chromosome number is 2n, and our chromosome number is 2n. This is just because of this meiosis. Meiosis is responsible for maintaining this chromosome number in a successive generation. Because when a next generation is born, for that the two parents have to contribute the gametes. They are haploid. So when two cells fuse to make a one cell for the next generation, the zygote, that becomes a diploid, and that's how that diploid cell makes a diploid body of the organism. Now, hypothetically, if you imagine that if there is no meiosis, an organism itself is which one? Diploid. Now, a diploid organism, if there is no meiosis, please listen carefully. If there is no meiosis, so the diploid organism makes gametes. What type of gametes will be there? Haploid or diploid? Tell me. They will be diploid. So एक diploid sperm होगा और एक diploid ovum होगा. उनके फ्यूजन से क्या बनेगा? What will be made by the fusion of these two cells? What's the name of that structure? Structure का name क्या होगा? Zygote. The structure will be what? Zygote. And the number of chromosome in that will be what? 4n. ऐसे दो diploid organisms जिसमें meiosis नहीं होता है, their gametes, the diploid gametes produced by mitosis only, that will create a tetraploid zygote, और tetraploid zygote में से body बनता है, वो भी tetraploid रहेगा. So that means species chromosome number change. It will be another species in the next generation itself. So what have you understood from this explanation? If one can unmute and give one line as a significance of meiosis. Meiosis is significant for dash 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 dash. एक line बोलना है. If you have understood this concept.
Chromosome number.
Lalo, are you speaking?
Yes, sir.
Be loud. It's not audible. Your
Sir.
every time it happens so. Hello.
Hello, sir. Now.
थोड़ा और यार, पता नहीं चल रहा है कभी.
Sir, away.
Yeah, it's fine now.
Sir, to see to that the chromosome numbers remain the same.
थोड़ा और explain करो. Means chromosome number same in what?
In this species, all the species
Exactly.
which are.
Every species should have a fixed number of chromosome in its diploid state, 2n. Our 2n number is 46. Drosophila करके एक fly है, fruit fly है. Its chromosome number is 2n number is 8, for example. So literally every species has a fixed number of chromosomes. But more important is that it should be maintained in evolution, otherwise that species will lose its existence. So species को retain करने के लिए वो chromosome number maintain करना पड़ता है. वो maintain करने के लिए when the species reproduces sexually, when it will use gametes, gametes में क्या होता है? Two cells are required. One is a male and one is a female, which will make a one cell of the next generation body. Because two cells have to meet, they cannot afford to keep them diploid. So they have to reduce their chromosome number to haploid, which is done only by meiosis, from the diploid to haploid. देखो माइटोसिस भी हैप्लॉइड प्रोड्यूस कर सकता है, बट वो हैप्लॉइड से हैप्लॉइड. हम क्या चाहते हैं कि डिप्लॉइड ऑर्गेनिज्म हैप्लॉइड गैमेट्स प्रोड्यूस करें, एंड दैट इज पॉसिबल ओनली बाय व्हिच वे? meiosis. This will help to retain chromosome number in a normal state. This is the importance of meiosis. The second point, during meiosis, one major important event occurs. That event, wait on. One very important event that occurs during the meiosis, that is crossing over. Does it happen in meiosis 1 or meiosis 2? Meiosis 1. Does it happen in prophase or metaphase or anaphase or telophase? Prophase 1. In which subphase of prophase 1 it occurs? Can you tell me? Crossing over. Yes, you may leave. Pachytene. So one of the most important event called crossing over occurs during pachytene, the exchange of segments by the homologous chromosomes at their non-sister chromatids. This will lead to a process called as recombination. New combinations of genes on the chromatids. एक क्रोमैटिड के ऊपर एक पीस मदर साइड से, एक पीस फादर साइड से. इसका मीनिंग एक नया कॉम्बिनेशन क्रिएट होता है जीन्स का. अगर ऐसे क्रोमैटिड्स आते हैं through sperm or ovum into the offspring. उसमें दोनों मैटरनल और पैटर्नल दोनों साइड का कैरेक्टर्स का एक ब्लेंड मिलेगा, and this will lead to what is called as variations in characters. New combinations of characters will be apparent over there, and that is something which is necessary in process of evolution. Have you all understood? मियोसिस जब जब होगा तब गैमेट्स में क्या होगा, गैमेट फॉर्मेशन के टाइम पे सेल्स में क्या होगा, क्रॉसिंग ओवर टाइम will lead to new combinations of chromatids. These combinations are passed through the gametes into offsprings. So offsprings exhibit what? Variations, which is a welcome phenomenon. Genetic variability is always welcome phenomenon in the process of evolution. Process of evolution, it is necessary. So that is another most important significant aspect of that of this. देखिए मियोसिस, बाय द वे देखिए, मियोसिस का एंड है, मियोसिस 2 का एंड है। आप देखो एक सेल है, जैसे ये स्पर्म बनेगा हाइपोथेटिकली, तो उसमें देखो एक क्रोमैटिड फादर का है, तो दूसरे में फादर और मदर के क्रोमैटिड का कॉम्बी है। इसमें एक मदर का क्रोमैटिड है, तो इसमें फादर मदर के क्रोमैटिड का कॉम्बी है। सो ये अगर फादर का गैमेट्स बन रहा है, मेल गैमेट्स, तो मेल के पैटर्नल एंड मैटरनल क्रोमोसोम है, मींस फादर के मम्मी पापा के क्रोमोसोम्स का कॉम्बी है। वैसा मदर के ओवम फॉर्मेशन में उसके मम्मी पापा का, मींस आपके कुछ जीन्स आपके नाना नानी की ओर से और कुछ जीन्स आपके दादा दादी की ओर से, मैटरनल ग्रैंड पेरेंट्स की ओर से और पैटर्नल ग्रैंड पेरेंट की ओर से। दैट इज व्हाट कम्स इन दिस, यू नो, जैसे नाना और नानी या दादा दादी है, ओके, ये पैटर्नल ग्रैंड पेरेंट्स है। फिर यहाँ पे मैटरनल ग्रैंड पेरेंट्स का अगर कंसिडरेशन किया जाए, उनके क्रोमैटिड्स मिलके आते हैं आपके और हमारे जैसे पेरेंट्स में। सो लेट्स से कि ये हमारे जैसे कोई मेल पेरेंट है और इधर हमारे फीमेल पेरेंट है, तो their chromatids, the gametes that are coming na, they themselves are coming with the chromatid combination of their mother and father, और इसके बाद हम आते हैं as the offsprings, हमारा जो भी जेंडर हो irrespective of that, हमें क्रोमैटिड्स इन दोनों में से मिलते हैं, हमारे मेल और फीमेल पैरामीटर, लेकिन उनके गैमेट्स में इन दोनों के भी क्रोमैटिड्स आ गए हैं। सो एक्चुअली वी आर like our grandparents, paternal side as well as maternal side. That is the thing which you need to understand, why lot of variability in us, though we are like, जैसे हम कितने एक ही पेरेंट के अगर दो सिबलिंग है फिर भी हमें इतनी वेरिएबिलिटी क्यों रहती है? It is just because that so many different combinations are created in the parental gametes that prepare us. So we carry some features of our grandparents of paternal side and maternal side. हम 4 different family के genes derive कर रहे हैं। अगर आप में इतनी सेंस से समझ में आया तो एक चीज आप अंडरस्टैंड कर लो, हम 4 different family, our maternal grandparent, नाना नानी, and our paternal grandparent, दादा दादी, तो 4 पर्सन के गैमेट्स दो में आते, जीन्स दो में आते हैं और वो उन दोनों के थ्रू वो हम में आते हैं, इसलिए इतना सारा वेरिएशन होता है। और ये ब्यूटी है मियोसिस का, that it creates so much of variation over there. Have you all understood this phenomenon?
Hello, sir.
हाँ बोलिए.
Sir, what if the variation turn out to be drastic?
एक लिमिट से ज्यादा नहीं हो सकता है, जो ड्रास्टिक का सवाल नहीं है. अगर इसी पैटर्न में इनहेरिटेंस हुआ है तो देयर विल बी अ सिमिलरिटी एज वेल एज सम वेरिएशन, बट इट विल नॉट बी ड्रास्टिक वेरिएशन अनलेस कि कहीं पे म्यूटेशन हुआ है तो ही. अदरवाइज अगर ये सीक्वेंस मेंटेन रहती है जीन्स जीन्स की पासिंग की, इसमें कोई अल्टरेशन नहीं आता है, तो फिर तो वेरिएशन जैसे हम दिखते हैं तो थोड़े बहुत तो हमारे नाना नानी दादा दादी इन सब से तो थोड़े तो मैच करते ही है. So it's a blend of matching as well as variation. It cannot be drastic variation unless the mutation or some other factor is interfering. Normal pattern में drastic variation हो नहीं सकता है. If they were human, our parents were human, grandparents were human, though we are supposed to be humans only, we won't have drastic differences in us.
Sir, could there be in animals?
Even in animal, pattern of inheritance is something which is following the same pattern in meiosis in every organism, so irrespective of the animal.
Okay, sir. Thank you.
चलिए, आप सबको क्लैरिटी मिल गया meiosis and its significance का. In bottom line, if we say it is necessary to maintain chromosome number of a species and also to bring in variation for the evolution. So आप एक चीज ऑब्जर्व करो कि evolution के point of view से मल्टीसेल्युलर एनिमल्स, डिप्लॉयड एनिमल्स में ये एक इंटरेस्टिंग एंड बहुत ही इंपॉर्टेंट फिनोमिनन था मियोसिस. माइटोसिस से नहीं चल सकता था, इसीलिए इवोल्यूशन में क्या डेवलप हुआ, मियोसिस हैज इवॉल्व्ड in this multicellular diploid organisms. Well, we shall have a break for 5 minutes and then we resume our work. Till then, enjoy the break. Madam, stop the recording for a while. Myocytes. We are talking about the differences between mitosis and meiosis. First point of difference, mitosis occurs in somatic cells to produce somatic cells. Meiosis occurs in myocytes in order to produce gametic cells. Remember. Second point of difference between them, DNA replication is followed by single round of cell division in mitosis. DNA replication occurs once and then there are 2 rounds of divisions in meiosis. मैं ये चीज आपको different way में explain करता हूँ. What they are trying to say. Have a look at this comparison of these two. See, G1 as G2M. Again, there will be G1 as G2M like that. Okay. In this process of mitosis, DNA replication happened and then after cell divided only one time. बाद में नया cycle start हो जाएगा. In another case, that is in meiosis, DNA replication occurred once and cell division occurred twice back to back. ये डिफरेंस आपको समझना है. अगर समझ में आया तो दोनों में डीएनए रेप्लिकेशन के बाद डिवीजन होता है. यहां पे एक बार होता है, यहां पे 2 बार होता है. That is the difference between mitosis and meiosis. And naturally, in this case, mitosis, a mother cell divides into two daughter cells. In case of meiosis, a mother cell divides and gives rise to four daughter cells. That is another difference. Then if you see carefully the chromosome number, mother cell and daughter cell chromosome number is same or different in mitosis. Chromosome
Same.
number in mother cell and daughter cells compared, is it same or different in mitosis? Same. Look at this. Chromosome number in mother cell and daughter cells, is it same or different? Different. Difference में कैसा है वो? Yes, half. We call it half the number of chromosomes in each daughter cell as compared to the mother cell. और जब भी ये क्रोमोसोम कंपैरिजन आए तो डॉटर सेल को किस मदर सेल के साथ कंपेयर किया जाता है? उसके जीवन स्टेज के साथ. याद रखना आपको, ये नहीं जो एंटर हो रहा था मियोसिस में या माइटोसिस में, ये जो जीवन स्टेज में है उसके साथ क्रोमोसोम कंपैरिजन ऑलवेज करनी चाहिए. रिमेंबर दिस. Then, as I mentioned earlier, one more point which I have already described. We'll go through the same. A diploid cell can go through mitosis as well as a haploid cell can go through mitosis. But in case of meiosis, it is only diploid cell which is capable of meiosis. Remember, meiosis can be performed only in diploid cell. Mitosis can be performed in diploid as well as haploid cell. Remember this. Further, synapses, crossing over, recombination, chiasmata formation. In which division these events occur? Mitosis or meiosis? Exactly, isn't it? So it is about major activities of meiosis which are comparison point. The prophase one itself, that is long, complex, important, significant one, with crossing over and recombination, all those, even synapses, chiasmata, all those are peculiarities represented in meiosis, not in mitosis. So that was another point of difference. And that lastly, the genetic material present in the daughter cells is same as in quantity and quality, same as in mother cell. For which division? Mitosis. Whereas genetic material in each daughter cell is quantitatively reduced to half compared with the mother cell, and in a quality it is divariant or with variation compared with the mother cell. So ये सब points of difference आएंगे. बाकी एक दो थोड़े थोड़े छोटे छोटे differences में तो ये आएगा कि जैसे माइटोसिस में single chromosomes arrange at equatorial plane, whereas in meiosis, particularly meiosis 1, pairs of homologous chromosomes get arranged on the equator. दूसरा, in mitosis, in anaphase, two chromatids separate from each chromosome. जबकि meiosis, particularly 1 में two homologous chromosomes separate from each pair. These are kind of differences which one has to understand, which exist between mitosis and meiosis. I give you 2-3 minutes to go through and raise your doubts regarding this. Well, as you are not raising your doubts, it means it is clear. Last topic almost. An unusual type of cell division, an unusual type of cell division that is seen in lower forms, bacteria, fungi, etc., protozoan, etc. Also in higher forms where the cells are undergoing degeneration. In these scenarios, in these scenarios, you can see one unusual type of division known as amitosis. It is called as amitosis over there. See, simply a parent cell first carries out karyokinesis, later carries out cytokinesis. Two daughter cells are formed. While this is going on, first of all, chromatin condensation event will not be seen. Second one, throughout the division, nuclear membrane will remain intact. So arrangement of chromosomes on an equatorial plane, spindle formation, all those things will not come into a picture. Remember that. These type of a primitive type of a cell division occurs in bacteria, fungi, even in some algae, and as I mentioned, degenerated plant cells. In all of them, such a division occurs. This unusual division is called as amitosis. It is known as amitosis. Is this clear to you all? Amitosis. Have any question on the same? It seems no question. Can take you to one more thing. Now, this is something what I am explaining to you will be useful for exam point of view. From exam point of view, you have to prepare this well. This concept regarding what happens to the chromosome number, what happens to the chromatids, etc., has to be learnt by you carefully. एक एग्जाम्पल है कि जैसे अगर सेल हैज अ 2C material in its mother cell, how much material, genetic material will be there in the daughter cell if it is going through mitosis? अभी हमने लर्न किया मदर सेल एंड डॉटर सेल जेनेटिक मटेरियल क्वांटिटी शुड बी सेम. इसकी जगह पे अगर अगर 4C लिखा होता तो यहाँ पे 8C हो जाता, 8C हो जाता और ये 4C हो जाता. I hope you have understood what I am trying to convey. Are you understanding the same? But if it was for uh, meiosis, the same question was for meiosis. Let's say somebody says that 2C is the genetic material volume in a mother cell. To always consider it at the G1 stage. So what will be the genetic material in the daughter cell? Tell me. यहाँ पे क्या आएगा? What will be the exactly 4C, 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 2C, 2C reductional division. और उसके बाद यहाँ पे CCC. This is the status of genetic material in the daughter cell if it is meiosis division. ये आपके माइंड में क्लैरिटी आनी चाहिए ये मैटर की, जो अभी मैंने आपको एक्सप्लेन किया. ओके, चलो कोई ऐसा बोलता है रिवर्स में आपको एक्सप्लेन करता. एक यूनिट है टू measure the to measure the volume, or rather the weight of genetic material like DNA. अगर वो पीजी में पिकोग्राम, उसको क्या कहते हैं, पिकोग्राम, किलोग्राम, ऐसा पिकोग्राम. So picogram is the unit to measure the weight of DNA. Very lightweight material like DNA, its weight can be measured in terms of picogram. Let's say that I would say that 5 picogram is the amount of genetic material, weight of the genetic material found in the gamete. Sperm या ovum, ऐसा कोई gamete है, उसका 5 पिकोग्राम वेट है. तो उसके मियोसाइट में कितना वेट होगा? Isn't it 10? रिवर्स जाएंगे, इससे इसका 10 पिकोग्राम होगा, ये वाला 20 पिकोग्राम होगा, ये भी 20, ये भी 20 पिकोग्राम होगा, तो ये 10 पिकोग्राम होगा. सिंपल. रिवर्स सोचो, किसी मियोसाइट में 20 पिकोग्राम genetic material है. So how much shall be there in its gamete? 10, ये भी, ये 40, ये भी 40, फिर इस डिवीजन में ये 20 और यहां पे आते 10 10 10 10 पिकोग्राम. तो दिस इज द वे यू कैन वर्क आउट द वॉल्यूम या वेट या फिर नंबर ऑफ क्रोमोसोम और क्रोमैटिड्स. ये सब कंसेप्ट्स जिसके बेस पे क्वेश्चंस आते हैं, you have to understand them. Have I made it clear to you all? चलिए, मैम ने आपको चेक पॉइंट्स के बारे में एक्सप्लेन किया, मैं एक बार पूछ लेता हूँ, क्लैरिटी मिल जाए हमें. Let's gather the information on checkpoints also one more time. So you know that in a cell cycle, there are checkpoints available. Normally कितने checkpoints होते हैं? 3. Who will define checkpoint? Who will define the checkpoint? Definition must have been given to you by ma'am. So you must have understood what are checkpoints. कौन define करेगा आप में से? अभी कुछ डिफरेंस के टाइम पे जो कुछ गर्ल्स ने बहुत अच्छे आंसर दिए थे उसमें से कोई ट्राई करे तो बेटर।
Sir, can I?
हु इज द वन?
Sir, Fatima.
हाँ जी, बोलिए आप।
Sir, it determines the exams or determines internal or external causes, whether to move it forward or not.
In whether to move it forward or not in?
Cell cycle.
Exactly. The points in eukaryotic cell cycle at which progression of a cell to the next stage in the cell cycle, that is been determined based on the conditions, internal and external conditions. If they are favorable, then only cell shall proceed forward in the cell cycle, otherwise it won't. These points at which it has been determined are known as checkpoints. Very good, Fatima. Who will describe about the first checkpoint, ch.10 checkpoint? Cell cycle started and towards the end of the ch.10 stage, a ch.10 checkpoint. Who will describe this checkpoint which occurs before the start of S phase? Nobody? Today all of you have given very good answers. Some of you should repeat. You people can manage actually. It's not always only Galav and Shreya and others. Others can also participate. No one is ready to speak about the first checkpoint.
Hello,
हाँ, कौन?
Sir Galav.
बोलो।
Sir, G1 checkpoint checks that the nutrients are present in the proper amount, growth factors are also there, and whether the DNA is not damaged.
DNA damage, okay. Basically, this is the checkpoint to decide whether the cell will complete the cell cycle, go for the DNA replication and cell division or not. Because here there is an option to go in that G0. So the cell has to decide whether it requires new cell. If yes, then cell division required. For that, DNA replication required. For that, it has to enter into which phase? S. So ये checkpoint decide करेगा कि cell अगर S में जाएगा तो DNA replicate करेगा. Replicate करेगा तो it has no choice but to go for division. This means that the cell goes for proliferation. So यहाँ पे आके अगर cyclins instruct करते हैं CDKs and cyclins कि आगे जाना है, that means conditions are favorable and there is a need for cell division. यहाँ पे आके अगर instructions मिलते हैं, दो possibilities है. Either condition cells required but conditions are not favorable, resources shortage, so do not go for the next further DNA replication. या फिर conditions may be favorable but there is no requirement for cell division, stop this thing. So cell चला जाएगा G0 में, depending whether it's a reversible or non reversible, वो उसके ऊपर depend करेगा, tissue के ऊपर. So are you all able to understand the restriction checkpoint G1 to decide whether the cell should go for next round of DNA replication and cell division or not? And that will be based on two aspects: resource availability for this activity, and second, the need for cell division. They will decide to go forward or not. G2 checkpoint. Who will describe
Sir, can
about
I
the
try?
checkpoint?
Shreya,
Sir, can I?
can I?
कौन है बॉयज में फर्स्ट?
Rudra, sir.
रुद्र, you try.
Yes, sir. G2 checkpoint mainly checks for the size of the cell, and it also have a watch on DNA replication.
Sorry, I could not hear properly. Sorry, if you can please repeat.
Yes, sir. Am I audible now?
Yes, yes, sir.
Yes, sir. G2 checkpoint mainly checks for the size of the cells and the DNA replication.
Okay. Further.
Sir, I have remembered in the question.
Fine, correct. So to check the size and mainly the DNA replication, priority is the DNA replication, G2 point. See, in S phase, DNA has replicated. And in G2, the cell has prepared for cell division. At this checkpoint, cell will decide whether entire DNA is fully replicated or not. And is there any mistake in the DNA replication? अगर आप इमेजिन करो कि यहाँ पे डीएनए रेप्लिकेशन में कोई मिस्टेक है या कोई अनरेप्लिकेटेड डीएनए, तो डिवीजन डिस्टर्ब हो जाएगा, ऑनकमिंग डिवीजन एम फेज. So here it is ensured that the DNA is properly replicated and completed its replication. दोनों चीज अलग होती है. रेप्लिकेशन कंप्लीट करना एक, एंड डैमेज डीएनए को रिपेयर करना या improperly replicated DNA को रिपेयर करना. Because that will make sure that equal type and quality and quantity wise equal type of genetic material will pass in the next daughter cells. So ये clarity करना जरूरी होता है second checkpoint पे, जिसको कहते हैं G2 checkpoint. It comes towards end of G2 just before entering in the mitosis. Who will describe about the third checkpoint?
Sir, can I try?
हाँ.
Sir, it checks that the spindles are properly attached to the chromosome.
This is called spindle checkpoint, correct? Also called M checkpoint, mitotic checkpoint. This will occur at the end of metaphase. Cell has just completed meta or almost about to complete metaphase and about to enter in the anaphase. उस juncture पे ये होता है. At this stage, it is ensured that all chromosomes have attachment with the spindle fibers. ये सेल है, यहाँ पे आपके क्रोमोसोम्स अरेंज्ड है इक्वेटर पे. And all chromosomes are aligned over there. Then only this checkpoint will allow the cell to go for anaphase. सोचो कोई एक क्रोमोसोम कहीं पे रह गया यहाँ पे, without attachment, without being brought at the equator. The cell will not go into anaphase. मैंने कॉल्चिसिन में भी एक्सप्लेन किया था, कॉल्चिसिन ऐड करते हैं, स्पिंडल फॉर्म नहीं होता है, क्रोमोसोम इक्वेटर पे अलाइन नहीं होते, तो एनाफेज प्रमोटिंग कॉम्प्लेक्स विल नॉट अलाउ द सेल टू गो फॉर एनाफेज. Because even if a single or all chromosomes not arranged on equatorial plane, not aligned over there, anaphase cannot proceed. Anaphase is about separating the two chromatids. कोई एक क्रोमोसोम यहाँ पे पड़ा है, अटैच ही नहीं हुआ, तो वो कैसे डिवाइड होगा फिर? Unequal chromatid number will pass on into the daughter cell, which is unacceptable. So this checkpoint will be crossed over only if all the sister chromatids are correctly attached to the spindle microtubules over there. Well, that was all about the chapter number 10. cell cycle and cell division. I have completed the same. Now, in the next lecture, we will begin another chapter. Hopefully, chapter number 6. I guess that's the one in the sequence left. So we shall start with that chapter.